Good morning. I'm Barbara Lohman, Board President of the Coastside Land Trust. We're so glad that you could join us for our free community webinar series this morning. At the Coastside Land Trust, we're dedicated to protecting the open spaces that define our coastline, ensuring they remain preserved for future generations. We focus on safeguarding scenic bluffs, stream corridors, and agricultural lands from Half Moon Bay to Montera. Some of our priorities include protecting land through purchases and conservation easements, securing funding for ongoing conservation efforts, helping landowners protect their land, leading local conservation, restoration, and educational programs. Our community is at the heart of everything we do, and we encourage you to get involved, whether through donations, volunteering, or spreading the word. Your support helps us protect the land we all love. Thank you again for being here today. And before I pass it on to Kate Dickey, our social media coordinator, I just want to mention that if you go onto our website, coastsidelandtrust.org, you will see all the past webinars and you may find some topics that you're curious about. So to introduce today's presenter, Kate.
Thanks, Barbara. And thank you all for joining us for this free webinar series. Our goal here is to bring together diverse environmental perspectives to help us to better understand the world and our role in caring for it, both locally and globally. Today, we were really excited to welcome Dr. Sarah Jacobs. She's the curator of botany at the California Academy of Sciences. Dr. Jacobs is going to explore the concept of species with a focus on the vibrant Castilleja paintbrush plants that thrive along our coast. As curator, Dr. Jacobs leads research on plant diversity, conservation, and ecological restoration. With a PhD in botany, she's contributed to numerous conservation efforts aimed at preserving rare and endangered species. Before we begin, a couple of quick reminders that if you have questions, we will love to answer them. Dr. Jacobs will be happy to answer these at the end. Please just type those into the Q&A section, and as we'll get to as many of those as we can in a formal Q&A session at the end of her talk. Dr. Jacobs, are you ready to get started?
I'm ready.
Thank you very much.
All right. Well, thank you everyone for being here, and thank you so much for the invitation to speak with you all today. I really do enjoy talking about Castilleja, and I love talking about species too. So it's just a real pleasure. I like to start my talks by sort of setting the stage of who I am as a scientist and the type of work that I do. I'm going to turn on a little pointer here. Okay, so I usually define myself when I meet somebody new. I tell them that I am a plant systematist and an evolutionary biologist. Most people are familiar with some of these terms, but systematist in general is one that is often new to folks. So I think it's helpful to see a definition. And so here I have sort of a smattering of different definitions of biodiversity science from across the years that I've sort of mashed together to describe what I do. And I think it's really nice to have this illustration here on the left of the screen to sort of put it into some context as a systematist as a whole. So first of all, I am interested in the study of the diversity of organisms. And this, I think, really relates to the role of taxonomy and classification for systematists. So we are the folks who put names on species, and we organize species into our biological classification system, which I'm going to talk about in just a few minutes. So that's one of the things that I do there at the top of that circle diagram. But I'm also really interested in how this wide diversity of organisms are related to one another, and how they're related to one another through time, so their evolution. This is the study of phylogeny, and we're going to see some phylogenies later in my talk. And so I'm also interested in the processes that generate all of this diversity and that tell us about how new species are formed. And so this is the study of the process of evolution, the nature and the origin of natural populations, both in the present and the past. But taxonomy has always been sort of my passion. It's what got me interested in plants and sort of what has directed my research through time. So our classification system, you've probably heard of it. We oftentimes learn some of these basic parts of our classification system in biology classes in high school and in our undergraduate. So I remember learning them as a mnemonic, kind of kingdom, phylum, class, order, family, genus, species. They are a series of sort of ways that we organize all of the diversity of life. And species are the smallest unit in this classification system. And this diagram here on the left shows you how each of these different levels of our classification system is nested one within the other. It's a hierarchical classification. At each level, organisms share characteristics. This is how we put them in their buckets of this is a species and these species belong to this genus, these genera belong to this family, etc. But the species unit, that smallest unit, really is one of the core parts of our classification system, certainly, but I think one of the core ways that we think about the world around us. And so it makes sense that we might take a minute to ask, why is the species rank so important? Why do we care so much about it? First of all, species are basically the cornerstone of the biological sciences. We use them in so many different ways. We use the term species to quantify and communicate about the diversity of organisms in the world around us. We say, California has this many species of plants, and North America has this many species of plants. We use them as a comparison and a contrasting tool. Species are also really important to the scientific theory and how we sort of work through the major components of the process of science. So every time we use a theory to make a prediction, or we design an experiment to test a prediction, we perform the experiment and then we interpret it and learn something and insert that back into our theory. Every time we go around this circle, oftentimes we're working with species, and those species are the units that we're considering as we're moving through this circle. And then finally, species have an incredibly important sort of social and cultural position with humans. They're one of the most important units that we consider for conservation. A great example of this, of course, is the Endangered Species Act. These are also units that we think of when we're managing and creating policy. So for all of these different reasons, species are super important to us. And for that reason, the way that we define species absolutely matters. We need to have robust estimates of species, and in order to use these units in all of these different ways that I've described. Now, defining species can be very easy. You know, here I've got a collection of pictures of all these different organisms that look very, very different from one another. But sometimes species can be extremely challenging to distinguish. So here I've got a collection of beetles. I'm a botanist, so if I use the wrong terms here, I apologize. But here we've got a collection of beetles that, from my perspective, look incredibly similar. But once we start peering in more closely, we start thinking about not only what they look like, but what their genetic material looks like, where they live, what they do in their day-to-day life. We might find that this collection of very similar looking things might actually be better represented by many different species. And actually, in this case, I think these are all different genera, so a different level of that classification system. So I think a next logical question, knowing that species can be very challenging to describe, is to ask, how do we decide what is and isn't a species? So in science, we love making definitions. And so in this world of species delimitation, our definitions are called species concepts. And then when you have a definition, you need some sort of rule to use to test if something meets the requirements of the definition. And so in species delimitation, we call this diagnostic criteria. So we have sets of concepts and diagnostic criteria that help us to determine if an individual belongs to a species using a specific type of concept or definition. So over the next few slides, I want to show you a couple of the species concepts that we frequently use, some of which I'm sure you all use. I'm going to show you how they work and then how they don't always work when we're trying to define what species are. So the first one is the biological species concept. And so the concept, the rule, is species are actually or potentially interbreeding populations that are reproductively isolated from other such groups. That's a big mouthful for sure, but basically the idea is if you can breed successfully, then you are members of the same species. And so here I've got a collection of pictures from the happy-faced spider, which is probably the best common name ever. And all of these spiders look very different from one another, but they successfully find each other, they successfully mate. And so under the biological species concept, we call these very different looking things members of the same species. Now, this particular concept has a hard time defining species when we don't know anything about their reproductive status or when they reproduce asexually. So a great example of a species that can't be diagnosed with the biological species concept is the paramecium that undergoes asexual reproduction. We also have a hard time applying this particular concept to organisms that we don't know much about their reproduction. So this is a case where this particular concept fails us. The next concept that I'll introduce is the morphological species concept. And this is a definition of species where they are the smallest groups that are consistently distinct and distinguishable. So just looking at their major morphology, what they look like when you look at them with the naked eye, the things that are consistently distinct are what we would call species. And so we might look at these butterflies here and determine where species are or where boundaries might be between species or not. But once we begin to understand that some of these species just represent variation within a species, like those top four, or have interesting life histories where they mimic one another, so they intentionally look very similar to one another in order to confuse predators, for example, the morphological species concept fails to determine where species boundaries are. So the butterflies are a great example of where this particular concept fails us. In the last several decades, as we have begun to learn more and more about the genomes of organisms, and we've peered into their genetics and looked at them from that perspective, we've begun to develop definitions of species that tell us how a genome evolves through time. And so here I'm talking about the phylogenetic species concept. So we use diagrams called phylogenies, and there's a fake example of a phylogeny here on the right of the screen. These are just diagrams that tell us how individuals, so these tips of the tree, how all of these individuals are related to one another. And so these tips are subtended by branches, and branches come together at places called nodes. And time moves along the axis here. So here at the tips is the present. And so all of these nodes and these branches go backwards through time. And so using these diagrams, we can see who is most closely related to one another. And here I've used some colors to help us see this. And then we can see who shares common ancestors. So this blue group and this orange group all share a common ancestor back in time. So under this particular concept, species are these terminal branches in phylogenies, or cladograms is another word for these diagrams. They're monophyletic, so they share a common ancestor here, and they're diagnosable by synapomorphies, so just shared characteristics. So this all seems very straightforward, but when we start looking at organisms or species that arise because of hybridization, so when two species come together and create a new one, sometimes this happens with many different species in the mix. We fail to be able to recognize species under this particular concept. So here, the diagram on the right of the screen is from one of my PhD lab mates. He studied a group of plants where hybridization was a huge driving force in the generation of new species. And so some of these tips that are colored in red, you can see they have all of these different colored lines that are contributing to them. Those are different lineages that have all come together in these hybridization events to create that species that's being recognized. And so in order to define species, he's had to use different concepts. So this is just a few of the concepts that are out there. There are actually 26, maybe even more, different species concepts that have been created through time as we have learned more nuanced parts of the story of how organisms change through time. I think that the biggest take-home message from this is that there's no single concept that we can really apply widely across the diversity of all organisms. And I think that's important to remember. If that is a goal that we have, and I think many people would argue that it would be good to do that, we haven't yet found a concept that can help us to define what species are across all known diversity. And, many evolutionary biologists these days are really moving away from this idea of a single type of process or a single definition of a species is what we need to work towards. Instead, we are thinking more about species as a process. So speciation is always happening. It doesn't stop. When a group of organisms are defined as a species, they don't sit statically and never change. They continue to evolve, they continue to change, and they continue to create new diversity. And so this continuous process of speciation is something that most of us are adhering to these days. And there's a really beautiful, simple illustration in some of the foundational papers of this idea. And in this idea, so time is moving up the diagram here, and you can imagine that at some point in time, there was one species, and maybe there were many populations of individuals that belonged to this species. But as time went by, different species criteria were acquired by these populations in different ways. And as time went by, more and more of these criteria were accumulated until at some point in time, groups of populations were very different from one another because they had these different criteria. And so when we think about when we're looking at species and trying to decide if they're species or not, we sort of need to understand when in this process we're observing. If we're seeing individuals at this time up here, we can easily distinguish them. But if they're still in these early stages of speciation, the gray zone of speciation, distinguishing species can be really, really difficult. So this process or this approach to thinking about species is, I think, being adhered to more and more by evolutionary biologists. It acknowledges that speciation is continuous, which is important. And it also underscores that in these early stages, this gray zone of speciation, any different criteria could be used to diagnose species. So we might see molecular changes that tell us where species boundaries are, but we could also use morphological criteria, what these species look like, or even characteristics from the environments that they inhabit. And so the job of the modern systematist like myself is to identify which of these criteria diagnose species, and again, when in this process we're sampling them. So, most of the questions or the way that I approach these questions in my particular system is, of course, thinking about this with respect to Castilleja. And, Castilleja is an amazingly beautiful, charismatic group of wildflowers. I think many people, when they think about Castilleja, think about it kind of like this. So this is a picture I took up in Canada during field work during my PhD. And there's these beautiful mountains in the background and this beautiful field of these bright red, beautiful flowers sort of, waving in the wind. And it really doesn't suck to be out in the field looking at plants when this is your scenery. But Castilleja come in so many different shapes and sizes and textures and colors. And so here I have a collection of photos that represent some of the different shapes and colors that we see across the genus. So there are about 200 named species in the group, and they're distributed widely across North America and South America. And this particular group, taxonomy and systematics, has been extremely challenging. It's been so challenging that folks have made jokes about it. They write about how difficult it is. And one of my favorite quotes comes from Harold Rickett, who wrote The Wildflowers of the United States. And this is from his Northeastern edition, where he says, "The genus Castilleja is one of those that make botanists wish they had embraced some easy branch of science, such as theoretical physics." It is a Western genus with at least 250 species, on the exact characteristics of which no two botanists seem able to agree. Fortunately for the readers of the present volume, only 4 species are found in the northeastern states, and they are not hard to distinguish. So this was a volume specific to that region where they didn't have to worry so much. Out here, west of the Rockies, it is tough to identify species. And so I find myself asking the question, like, why? Why is taxonomy in Castilleja so difficult? And what I've decided is that these traits that we use to distinguish species are largely overlapping. If you think of like a Venn diagram of different species, they're like totally over one another, overlapping. And this is especially the case for many of the traits that are outlined in the literature that we use to define species. So or to recognize species, like the Jepson flora or the field guides. And these criteria are morphology, the way that they look, the habitat that they belong to. So when those are the only traits that we have, it's really hard to tell them apart. So I want to talk a little bit more about these 2 particular traits to show you some of the breadth of variation in the group. So first, I want to think about habitat as a diagnostic character. So one of the reasons why the habitat is so difficult to use in Castilleja is because they occur everywhere. They have what we call a broad ecological amplitude. So we can find Castilleja in very wet areas like meadows, like Castilleja cusickii here. But you can also find Castilleja in really dry deserts. So this is Castilleja scabrida from the Great Basin. You can find them at low elevations. This is Castilleja latifolia here on our coast in California. But you can also find them in extremely high elevations in the alpine zones. So this is Castilleja nana in the White Mountains of the eastern Sierra Nevada. We even find Castilleja in extremely harsh environments. So this is Salsuginosa, which is found in these alkaline basins in Nevada. So they are pretty much found in all different types of habitats. We also have species that are very broadly distributed. They occur over these really big geographic spaces, but we also have some that are very narrowly restricted, very locally endemic. These are often species that we have conservation and questions around. But otherwise, they're literally almost everywhere. Probably they are almost everywhere because Castilleja are actually parasites. So I don't know how many of you know that Castilleja are parasites, and technically they're what we call a hemiparasite. So they're fully capable of performing photosynthesis. They can make their own food, but they can also form this really interesting organ called a haustorium. This is an organ at the root for Castilleja. And so here we've got a diagram of a close relative of Castilleja. This is the genus Striga. So Striga is here, and the host is here. And this organ that Striga has generated, it's produced, is called the haustorium. And here we've got a cross section of what that looks like if we did a longitudinal section. And so here we've got all of our parasitic tissue here, and then the host tissue here. And you can see that the cells of the vascular tissue of the parasite are literally punching into the vascular tissue of the host, and they're slurping out water and minerals and nutrients and all kinds of good stuff. So Castilleja do this as well. And here I've got some pictures from a colleague at the University of Wyoming. So, and so these are the haustoria of Castilleja specifically, these little nodules here. So this is a penstemon here in the center of this Petri dish, and these little baby Castillejas here on the right. Here the soil has been cleared away, and then we can get a really nice view of the haustoria. So we think that Castilleja are what we call entirely facultative, which means they don't absolutely have to have a host in order to get through their life cycle. In fact, I've seen many examples of Castilleja that appear to be growing straight out of the rocks. And it's hard to imagine where their host material or their host might be. And so it's possible that this particular individual is surviving without a host. So there's another reason that I haven't quite touched on yet about criteria that diagnose species that are difficult to apply in Castilleja. I mentioned it earlier with the genetics piece, and I just want to give you a quick insight about the genetics of Castilleja. The genome for the group is a complex sort of characteristic that we've looked at to try to define where species boundaries are, but the more we learn about it, the more complicated it gets. I'm not going to talk a lot about this today, but I did want to give you just a brief overview of it. So from a molecular perspective, the group is very complex. They're a young lineage. The common ancestor of Castilleja is only about 4 to 6 million years old, which of course sounds really old to us, but it's quite young. We regularly see evidence of gene flow when species co-occur, and so that makes understanding their genetics very difficult, kind of like the hybridization I was talking about earlier. So as an example, I've got when I was in the White Mountains several years ago, I found this really beautiful Castilleja linearifolia that has this sort of shape of flowers here. In that same population, I saw Castilleja pilosa, which has a very different looking set of floral organs. And in that same population, I saw these really beautiful intermediate plants that looked a little bit like linearifolia, but also a little bit like pilosa. So this is an example of these examples of gene flow that we can see in the field. The other thing that Castilleja does that makes genetics in the group really tough is that they're what we call polyploids. So they duplicate their genomes. And within a given individual, you might have 4, 6, 8, 10 copies of the genome. Polyploidy like that makes it very difficult to apply our genetic analyses. And so the genome as a diagnostic character is definitely something that we're all working towards, those of us that study Castilleja, but it isn't one that we have a really tight story for as of yet. So the last series of slides to introduce you to Castilleja is all about the morphology of the group. So what the flowers look like themselves. So, and of course, thinking about this in the sense of a diagnostic character for defining species. So the basic parts of a Castilleja are these. So here on the left, we have, a typical plant. At the tip of each of these branches is an inflorescence or a series of flowers. If you peel one of those flowers off and you break it down into its component pieces, you'll see the bract, the calyx, and the corolla. And it's really the calyx and the corolla, or the sepals and the petals, where we see the major morphological groups. And here are some outlines that show you what some of those shapes look like in Castilleja. So we think a lot about flower shape in the group because flower shape really determines how pollinators will interact with the flower. And so this is me using a probe to sort of interact with one of the long-beaked forms in the way that a hummingbird beak might interact. And you can see that as I push it into the beak of the flower, the anthers are sort of revealed. Just like that. Yeah. So historically, we have organized all of the known species based on these major morphologies. So here I've got some boxes under each of the shapes that tell you sort of the names of the sections and how many species have been described under each of these morphotypes. And if you look closely, you can see that several of these are represented by only a single species. So these are shapes that are fairly unusual amongst the group. But most of the rest of the species have really been defined under these particular 5 shapes. And we further sort of organize these based on how we think that pollinators engage with them. So those 2 photos or pictures on the right have really long beaks, and we think of those as being hummingbird pollinated, whereas the 3 on the left are kind of short beaked, and we think of more bees and wasps interacting with them. We also know that their life history habits, like whether they live just for a single year, like the annuals, or whether they live for many years, like the perennials, it varies across these morphotypes. And if we think about how we might start envisioning how these particular shapes and life history traits might be related to one another, we might suspect that perhaps these long-beaked versions that are both perennial are related, and these short-beaked versions that are both perennial would be related. And then given that all of these are perennial, we might suspect that they're more closely related, and then the annual ones might be related in this way. So this is sort of how we begin to arrange our knowledge and our understanding of organisms into these diagrams that tell us how they might be related to one another. And so once we have these hypotheses of these relationships, we can begin to classify them. So we ask, what are the species? How are they related? And how many are there? And then once we have that type of information, we can ask even bigger questions like what processes generated these species? What is the fate of species in the future? And in Castilleja, the taxonomic confusion, all of that confusion that people are writing about and joking about, really has to do with what are the species within these major morphotypes. And so when I approach my work in Castilleja, I'm often thinking about all of these species that have been named as hypotheses, and my job is to test them and try to refine our definition and our, our understanding of them. So when I started my job at the Cal Academy back in 2020, I initiated probably one of the most complex groups, one of these morpho groups or these species complexes, and started gathering data here. And so the last part of my talk, I'd like to introduce you to what I am calling the California Coastal Castilleja Complex, which is a huge mouthful. A friend of mine suggested that I call it the C4 complex, which rolls off the tongue so much better. But I really do think that this is very much the epitome of confusion in Castilleja. It really has, this group of species has all of the different elements of what make distinguishing species so stinking hard. So here I've got a picture of each of the 5 sort of members, core members of this alliance. And here on the right, I have, a map of California, and each of those dots is a different location where we've collected a specimen that are housed in herbaria, and that's where I work at the Cal Academy. And they're color coded according to the species identification that botanist gave them. So I very purposefully did not tell you which of these pictures goes with each of these colors, but because frankly, I'm very suspicious of species, as I think I've said. But this does give you an example of, or a visualization of how it's been hypothesized that these species exist across the coast of California. And you'll notice that some of these colors have very distinct geographic ranges. For example, the red ones, they're in sort of Northern California, the yellow, the green, and the blue. Purple is pretty widespread. But each of these more restricted things is usually a species that we are thinking about when we talk about conservation and the management and trying to protect species, right? And so here again, we're getting into this discussion of, are these actually species? Is this where we should put our resources, as a more sort of applied question to my research? Okay, so these are the core members of the Alliance. Now, as I started gathering data for this particular group, I found that there's actually additional red perennial Castilleja that completely overlap in ranges of these core species. And as these pictures illustrate, they look extremely similar. So really, I think this complex is more like 9 species, potentially more, that we're really having to sort of try to tease apart. And ultimately, these taxa are just super hard to identify, especially in those areas where their geographic ranges overlap. Another thing that makes it really tough to identify them is that each of these core species has a morphological variant. So basically, there have been other populations of these species with different colors and other sort of nuanced characters that are very restricted that add another element of difficulty distinguishing them. And in some places, when you visit populations of these species, you'll find extremely different shapes and colors in a single population. So this is from an area close to Point Reyes, not far from me here in Marin. And this was all within maybe like 10 meters squared. I saw each of these different shapes and colors. And so every time we go out to look at these species, variation is very much expected, both across populations, like we see in the columns above, but also within populations. And I'm beginning to wonder if we even see variation on a single individual. And then the last part that makes, distinguishing species in this group so challenging is that most of our keys, like the Jepson flora or the flora of North America, they ask us to use habitat characteristics as a way to distinguish species as well. But when we look at the habitat characteristics for these species, we find how incredibly overlapping they are. They're all along the coast of California. And so here I have the 5 core species listed, and then each of them has, the habitat descriptors of where that species belongs. And then in the next few slides, you're going to see a bolded red color of habitat characteristics that are similar. So here, if we are standing in the chaparral along the coast of California and we encounter one of these things, it could be either Castilleja affinis or Castilleja latifolia. If we're standing on a sandy bluff or a sea cliff, we could be looking at latifolia or littoralis or mendocinensis or ydii. If we're standing in grasslands, Who knows? And I mean, if we're in coastal dunes and scrub, then it's basically impossible to tell them apart. So, ultimately, we know that there are different things here, but we can't seem to quantify it. And it's inconsistent across habitats. And so my team at the Academy has really been trying to figure out ways to tackle this problem. So Dr. Adriana Hernandez and Magdalene Lowe were both working with me in 2023 to tackle these problems. They've both moved on to their next career phases. So right now it's just me, but we developed a field survey app to collect ecological data for every individual that we encounter in the field. We also take scans of plants. We do dissections and put them on a scanner and take a picture of them. And then we do some really fancy morphological measurements to look at the shape of floral structures. We're also looking at other elements of the group of the individual that could help us define species. So Magdalene started digging into what the pollen looks like of plants. And then, of course, we're gathering molecular data. And this is much more nuanced work that is going to be very exciting, but much of that data is not yet collected. So what I would love to do right now is to share with you some of our preliminary morphological data from our 2023 field season. And this involved an REU intern, Lily Case, who was absolutely instrumental in helping us to generate this data. So what I'm going to show you are some basic linear measurements, so just length and width of various floral features from 100 different individuals that we collected this first season. And basically all I'm going to ask is how well do these individuals adhere to or fit within the expectations of what we think these species should look like? And I'm going to say, is there any morphological variation in this group? And so what this is morphology in light of current taxonomy. So taking the current species definitions and trying to understand if the morphology that we can see in individuals fits within that expectation. So these are some slides of the basic parts of Castilleja, just across a handful of individuals that we sampled. And again, I'm going to ask about what does morphology look like in the light of current taxonomy? So here I have some box plots of some of our raw measurements. So we have measurements associated with each of the main floral features, the bract, the calyx, and the corolla. And then each of these sets of plots are the measurements for a particular linear measurement, and they're organized by species. So each of these colors corresponds to a different named species. And you can see as we look across each of these individual measurements that in some cases there's a lot of overlap in the measured trait. So here we can see that these boxes are all together. But sometimes there is some variation. So over here in the corolla, we can see that Mendes sinensis really does have a very different sort of measurement relative to most of the other ones. Now, we can think about the expectations when we read the Flora of North America or we read the Jepson Manual, we have an expectation that these traits have a certain length and width. And so here what I've done is taken just this particular trait and I've outlined the minimum and maximum measurements that we expect for these individuals. And then the box plots are the raw data from the samples that we gathered. And so you can see that for many of these species, so each of these box plots is a different species, most of the measurements that we've taken fall well outside of the expected measurements. And so this is just an example of how we use some literature to try to understand what a species belongs to, and our measurements are not matching it, which is disconcerting. And we see this across different organs and different types of measurements. And so the next type or another way to think about this is if we look at the expected length of one of the flower parts, the corolla, for each of these species, this is what it might look like. So this red line indicates that we expect the length of the corolla to be between 15 millimeters and 40 millimeters long for Castilleja affinis. But this is also the length of all of the other species for that particular feature. And so you see a bunch of the overlap just within this particular measurement. So if we measure individuals using just corolla length, yes, they fit within Castilleja affinis, but some of them also fit within latifolia and littoralis and mendecinensis and whitei. Now, if we add a second characteristic and make this what we call a biplot, so we consider the beak length, we can create these boxes that sort of illustrate, like, given a certain corolla length and a certain beak length, what are the ranges of measurements for an individual? And so here are individuals of affinis that have both of these traits, and you can see that they fall within our affinis box, but they also fall within the whitei box and the latifolia box and the littoralis box. And so here are individuals defined as latifolia. Here are some as littoralis, mendecinensis, whitei. And so the point here is that all of these fall within all of the boxes. And so how can we possibly decide what species are using these two particular traits? So the last tool, the last arsenal that we have to sort of explore this is to think about what we call principal coordinates analysis or principal components analysis. And this is where we consider all of the different traits at once, and we create what we call a morphospace using all of those traits. And when we plot individuals into this morphospace, we look for them to occupy different areas if they're morphologically very distinct from one another. And as you can see with all of these overlapping circles and these overlapping colors, there's just not a lot of distinction. And so I ask myself, is this a common pattern in other species complexes in Castilleja? And the answer is yes. Yes, it is. So this is Pilosa and Nana that I worked on during my PhD, and Castilleja cusickii I also worked on during my PhD. And these are cases where these complexes are more or less distinct until they co-occur with one another. And morphology is poorly diagnostic. So when we bring it back to the end and we ask, what does it mean to be a species in Castilleja? And from a morphological perspective, what I think it means is that we're recognizing all kinds of variation when we're out in the world, but maybe we're a little too quick to put names on them. And so when we test these species, when we try to better understand these species, we really need to consider a lot of additional things, not just what they look like. So how does the host play into where species are? What about where they live? And some of the genetic components of these individuals. Now, I didn't talk a ton about the molecular perspective, but it is important to say that when we attack the idea of species using the genetics, we really do need to be careful. So they have a very complicated molecular history. They engage in hybridization, and really, we just got so much work to do with the genetics piece of it. We need to create new resources, of course. We've got to be thinking about how many copies of their genomes they have. Ultimately, though, I do think that this really sort of simplified what we call a bifurcating relationship like this might be an inappropriate sort of thing to be working towards. When I think of species in Castilleja now, I think more about braided river channels. And I think this is a better description of what species in Castilleja might be. We've got populations that diverge from one another, but they come back together in hybridization events only to create even new types of species as they go by. So this is what we call a network model of relationships. So we stand to learn so much about the early stages of speciation by studying groups like Castilleja. And they really do bring in all of the major components of the systematists' work. And then, of course, they also make these really beautiful herbarium specimens. And then just as a last note, I know I'm a few minutes over here, but I think it also stands we we stand to gain a lot by asking, what does this mean about species more broadly, like outside of Castilleja? And, this has been an enduring question in evolutionary biology, and I suspect it will be for a long time to come. There's a lot of us who just sit here and we rub our head because sometimes it gets a little painful to think about it. But ultimately, it's important to remember that these things that we call species, they're absolutely a human construct. And at any point in time, they are definitely a hypothesis to be tested. And so that is what I will continue to do at the Cal Academy. So I want to say thank you for inviting me to talk. I'm so sorry I went over by a few minutes. Thanks for hanging with me. Thank you very much for your attention, and please do wish us luck. I think we're going to need it.
Oh, we do. We wish you a lot of luck, and we're here for you. We have a couple of questions just before we go. So Mary was asking, just because this is what you were just speaking to. She said this is a simplified question, but are they capable of, if they're capable of breeding fertile offspring, why not just call them one species that's highly varied?
Yeah, I mean, I definitely think that that's where we're headed. The only caveat to that is if there are, there are these types of species called syngamians. Oh, I kind of wish I'd remembered to talk about this, but and what they are things that look morphologically very different, and genetically they appear really similar, but when you have huge amounts of genetic data and you can look at like every single base pair that exists in the genome, sometimes you can find these few variations that correspond to these different morphologies. And there's a hypothesis that this is sort of an approach that lineages will take to sort of, they retain their ability to exchange genes with one another so that they can survive really well in the habitat that they find themselves in, especially when that habitat is extreme. So if you think about yourself as being like really suited to, the dunes, but you're able to exchange genes with some, another species that lives a little higher up in elevation, maybe in drier areas or different types of soils. If you exchange genes with them, maybe you will get some beneficial genes that will help you in your particular location. Or if your location is experiencing a lot of tough times, like extra drought, or as the climate is changing, just changing environments. And so in those cases, if we go ahead and still recognize them as different species, we capture the existence of a syngamian. And so while it might be the easiest thing to just call them all a single species that has a lot of variation, we still do need to make sure that it is the most appropriate way to define them. And again, it's like we're learning about the process of diversification, of speciation, by recognizing those types of things.
Mm-hmm. Mm-hmm. Okay, Larry asks, is morphology as a diagnostic tool losing validity as molecular knowledge increases?
Oh, that's a great question. So I do think, in a loose way, I think most people got super excited about genetics, and many research programs really did sort of, I wouldn't say that they abandoned morphology, but they really did like a hard pivot and really poured a lot of time and resources into molecules and genetics. And we started learning some really amazing things. For example, how when we look at the genome, it tells us something different sometimes than it does with morphology. But I do think, so we've been doing this for like 3 or 4 decades now, and we're learning so much more about genomic evolution and how genomes change through time, and our ability to detect these changes and to analyze them and to model the processes associated with genome evolution. And we're realizing that it's not the answer to everything. We can't forget about the morphology and the ecological traits and all of the other elements associated with an organism. And so I don't think that we ever actually forgot it, and I don't think that morphology is losing validity, but I do think we've sort of been like hyper focused in the genome, but we're really beginning to shift back into a more holistic consideration of what species are.
Mhm.
Oh, wait, one more thing I'll say about that. You know, we also need to be thinking about the utility of species. You know, like we don't have little handheld devices that we can take out into the field and like, you know, scan something and look at its genome. We are still restricted to observing organisms in the field as they look where they are. And so it is very useful for us to have these morphological features that we can use to define species. But I do think we're going to have to come to terms with being out there and saying, you know, that looks like Castilleja affinis, but unless we looked at its genome, it could also be this other species. Yeah.
Mhm.
Connection to being out in the field with the naked eye, but your field survey app
Mhm.
that you created, is that available to citizens?
So it is not really, no. It's designed specifically for Castilleja, but it was built using software called Survey 123, and that is in the framework of the software called ArcGIS. And it's a pretty easy sort of software to implement. It's not that difficult to make a basic one that you convert into this app and install on your phone. Um, if somebody's really interested in sort of diving into this and making an app for themselves, please email me and I can point you in the direction for how we learned how to do it. But yeah, at this point, no, I haven't published it for broader use.
One of our, I'm sure you know, Tony Corelli, our local botanist, and she asks if you can talk about your work in conservation of Castilleja.
Yeah, so you know, I've been largely involved in trying to understand the processes that are generating this diversity. Much of it hasn't yet been applied to, um, you know, actual policy or management plans that have been enacted by folks who are doing that type of work. Um, Now, my work in my PhD did sort of provide some of the genetic data that would elevate some species to species status, or they were called varieties before. But that hasn't been formally done yet, in large part because this molecular backdrop is yet to be sort of fully fleshed out. However, since I've been at Cal Academy, I have engaged in a couple of collaborations where we're beginning to develop that very closely. So we have a new project starting with the Tiburon Paintbrush, which is Castilleja affinis subspecies neglecta. And this is going to be a project that builds a lot of genetic data to ask just within this subspecies, how much genetic diversity do we have? And what does that tell us about how we should best manage? And this is a collaboration with Creekside Science team, which are the boots on the ground that are actually doing the applied work. And so this is, I think, where my work has the closest intersection with conservation, is by providing the data and sort of the analytical perspective that is then worked into conservation plans.
Okay, another jag here, but
Okay.
and any evidence that epistatic genes are controlling the variation?
Oh, that's amazing. That's a fantastic question. That's definitely a person who knows some good genetic stuff. I don't know that we have any evidence for epistatic interactions. My brain has been mostly thinking about what are the roles of the host and what the phenotype looks like. So if you're a Castilleja attached to a lupin, do you look a little different than if you were attached to a grass or a shrub like Baccharis, for example? What is the role of the host? Also, what is the role of the substrate? So we do have some species that have been named that are endemic to serpentine outcrops. Usually those are different in color and different in stature. These are serpentine areas are really nutrient poor areas. And so what does the role of that local environment do in terms of shaping the shape and the color and the stature of the plant? That's where my brain has really been spinning about the phenotypic variation. I also think that hybridization is contributing to that too. And so I think there's a lot of different roles, but it wouldn't surprise me at all if there were epistatic interactions as well.
All right. Well, I want to value your time because as many of you probably who know Dr. Jacobs, she is part of this core team that is curating over, it's like 2.5 million herbarium specimens at the Cal Academy, and doing this incredible research and then also making time to speak with us. And so we are so grateful that you've made this time to speak with our population here, our community here, and share not only just so much about the process of speciation and your, all of this, the Castilleja, we're going to be looking at it in such a different way, but also just your passion. It's palpable, your passion for this.
Oh,
and
thanks.
we're so thankful that you made this time.
Thank you for having me. It was an absolute pleasure.
And the rest, for those of you who are sticking around, we have, as many of you know, we will be sending a follow-up email. So I know someone sent an email or a question saying, can you give us a quick synopsis? A recording will be coming to your inbox tomorrow, and that'll give you, you can please share the recording. And as Barbara Lohman shared at the beginning, if you hop on our website, it's a wonderful opportunity to get back there and look at just check out some of the past webinars that we've had. Just a chance for this rich broth of these incredible, esteemed presenters sharing about what's going on locally and things that so many of us are just learning fresh through these eyes of people that are really, really established. We'll say also, we have a couple of things coming up. So just in addition to the follow-up email, there'll also be a link to our events page. Take a look because we do have a couple of things coming up. Really locally, we have the high school art show, and that will be coming up on February 7th. And that's during that first Friday. So it was interesting that Janelle Bracamont of the Half Moon Bay High has challenged students to make these original compositions, including they had to kind of randomly pick a subject, an activity, and a place and come together. And they've created these really cool compositions. So there'll be a gallery opening on February 7th from 4 to 6. Call out, of course, always that second Saturday of every month. We have a core team that we're really hoping to grow of people that are want to come in and dig in to some of our restoration projects. And some of that is weed removal, some of that is greater, restoration efforts. But if you are interested in getting involved in kind of a deeper way, please reach out. Again, you can hop on our website and get that, but otherwise just info@coastsidelandtrust.org. And then we have Timothy Federal from San Mateo Resource Conservation District will be coming on as a webinar in March to talk about wildfire management, habitat restoration, and climate resilience on the coast. So lots to look forward to. Thank you again, Dr. Jacobs, for being here. Thank you all for committing to learning more about our local environment, and we look forward to seeing and hearing from many of you soon. Thank you.